Good morning, everyone. We are good too, and today we are super excited to share with you something different. Tillandsias, also known as airplanes. Now, we think of planes. We usually think of roots in soil, right? But airplane doesn't need soil at all. They grow by attaching themselves to trees, grass, or even power lines, just hanging out in the air. That's because they are what we call amphibians. Plants that live on other surfaces but don't take nutrients from them. You may be surprised to know that Tillandsia and pineapple actually come from the same plant family, Ruminaceae, but they are very different. Pineapple grow in soil and give us fruits. Airplanes, on the other hand, are grown for decoration and survive just by absorbing moisture from the air. And airplanes come from Central and South America, and also parts of the southern United States. And they are super adaptable. Some grow in rainforests where it is really humid, while others survive in dry deserts. Since they don't need soil, they just grab onto tree barks, grass, or fences, and get everything they need from airflow and humidity around them.
Unlike most plants that rely on their roots for water uptake, Tillandsia is an exception. Instead, it captures water directly from the air using the white structure on its leaf surface called trichome. A trichome is made up of several layers of cells, the wing cell and shell cell, increasing the surface area to catch fog droplets. Water then flows downward through the down cells and into the food cell. These food cells have a semi-permeable membrane, like a selective gate for water. On the side, a cuticle will prevent the water from leaking out. Finally, water reaches the hydronkima cells. These hydronkima cells don't just store water, they help put it in, maintaining a strong negative water potential around minus 1.2 megapascals. Compared to the water potential outside of the leaf on the foggy mornings, which is close to zero. That is when trichome get to work. The outer shell cell capture droplets, and water flows inward from the air through the down cell and food cell and into the hydronkima. Now here is the problem. When the environment gets dry, the water potential outside drops to minus 100. Water could start leaking out. But Tillandsia has a solution. With just a little evaporation, the air-liquid interface begins to shift inward, from the outer shell into the trichome's inner structure. This is the valve switch you can see in this diagram. From the left to the right, once the water surface moves deeper inside, liquid water can't escape anymore. Instead, it has to leave very slowly as a vapor, passing through the thick shell cells. This unidirectional water valve makes Tillandsia one of the few plants that can survive in fog deserts and extreme dry zones.
Although Tillandsia doesn't absorb water through its roots, it still requires essential nutrients to grow. The three key micronutrients, nitrogen, phosphorus, and potassium, each play a vital role. Nitrogen supports leafy growth and tissue development. Phosphorus helps with energy transfer and root formation, and potassium regulates stomata and stresses stress resistance. For air plants, these nutrients are typically absorbed through foliar feeding, spraying them directly onto the leaves. Growing Tillandsia isn't about soil, it's about recreating the right environment. First is water. Regular misting mimics dew and fog, the natural sources of hydration. Next is airflow. Good ventilation helps dry the leaves, prevent rot, and may serve as a mechanical cue for roots or leaf responses. Finally, sunlight. Most Tillandsia species prefer bright, indirect light. Light lasers not only influence photosynthesis, but also affect trichome development and density. Together, these three, moisture, airflow, and light, are the keys to keeping your air plants happy and healthy.
Tillandsia reproduces both sexually and asexually. Sexually, it produces flowers that combine both male and female organs. Each flower typically has 6 yellow stamens and a central white stigma. These reproductive structures has arranged within purple tubular petals, creating a striking contrast that attracts pollinators such as bees, moths, and even hummingbirds in some species. After pollination, seeds develop in ovary. These seeds are equipped with fine, sticky hairs called comas, allowing them to be dispersed by wind over long distance, ideal for colonizing new aerial habitats. Asexually, they produce pups, clonal offspring that grow at the base of the mother plant. By combining seed space dispersal and vegetative cloning, Tillandsia achieves both genetic diversity and rapid local spread, helping it survive and thrive in harsh or changing environments.
Now I'm going to talk about our experiment. The first experiment, investigating root adhesion of Tillandsia under different growth conditions. In this study, we wanted to explore how different environments might affect the plant's ability to develop roots and adhere to surfaces. We hypothesized that Tillandsia would develop different root structures depending on their environment. In soil, we expected longer and more branched roots, but weaker adhesion. In aerial conditions, we predict root growth will be minimal and finer, as the plant isn't in contact with any surface. On bark, we expected stronger adhesion due to potential secretion of adhesive substance. Our aims were threefold. First, to evaluate the root growth patterns of Tillandsia under different conditions. Second, to assess their ability to adhere to different surfaces. And third, to investigate how environmental factors such as substrate and exposure influence root development. Cultivate plants under three conditions. Conditions in air, mounted on bark, and potted in soil. After 2 months of growth, we record the root status of each sample in all three groups. Here, we show photos of the sample for each group. As you can see, root development was visually limited, regardless of condition. Only one plant in each treatment group developed roots at all. Each of those plants produced just a single root, with no branching. Overall, root development was limited, and we found no significant difference in adhesion strength between the groups. Therefore, we could not confirm our initial hypothesis with the 2-month experimental period. We identified two main possible reasons for the limited outcome. The environmental conditions were too stable. In nature, Tillandsia is often exposed to wind, rainfall, and movement, stimuli that were absent in our lab setup. Second, the observation period may have been too short. A 2-month cultivation period likely wasn't long enough to induce clear differences in root behavior. Even though the results weren't as we expected, the process helped us reflect on what Tillandsia needs to grow roots. Now, let me show you how do we improve our experiment.
Tillandsias. Okay.
Although our roots experiment was unsuccessful, the roots barely grew. We suspect the problem wasn't the plants, but the lack of environmental stimulation. First, when the environment starts getting turbulent, wind, shaking, instability, the Tillandsia realizes, okay, time to grow some roots. Next time, we'll add real pressure, stronger wind, simulated rainfall, and a longer growth period.
Welcome to our second experiment. In this study, we explored how light intensity affects the development of trichomes in Tillandsia. These leaf structures play a key role in water absorption and protection. But how does light influence their growth? We hypothesize that light intensity has a strong influence on trichome development. Under high light conditions, plants would develop more and denser trichomes to enhance water absorption and minimize water loss. Under low light, the need of such adaptations would be decreased, leading to fewer and less development trichomes. To test this, we set out two objectives. First, to quantify the number of trichomes under different light durations. Second, to observe how their morphology changes under the microscope. Would their structure remain the same, or would the plant allocate resources differently? Here are our raw results. We test 3 different photo periods, 6 hours, 9 hours, and 12 hours of light per day. For each group, we took microscope images and counted trichomes in a fixed unit area. The 12-hour group showed the highest counts, followed by 9 hours and then 6 hours. There were some variations, but a trend was starting to form. To visualize this, we plotted the average trichome counts for each group. As the light duration increased, so did the number of trichomes. This supports our hypothesis. More light seems to trigger denser trichomes development. Let's analyze this more closely. The highest light exposure resulted in the most trichomes. The lowest light has the fewest. This consistent trend across all groups reinforces our hypothesis, suggesting that the Tillandsia adjusts its leaf structures based on environmental light cues. So why would this happen? Trichomes likely serve a dual function. First, they help absorb and retain moisture. Second, they act as a protective layer against water loss. More sunlight means more evaporation, and thus more need for this natural protection. Here is a note. Some microscope images were slightly unclear, which could have introduced minor variation in the counts. For the discussion, there are one limitation. We only measured trichome density at the base of the leaf. This may not reflect the full picture, since different parts of the leaf receive different light exposure. In future studies, we recommend multi-point sampling to better understand how trichomes adapt across the whole leaf surface. This is the end of our presentation. Thank you for watching. We hope the introduction of the airplane and these experiments helps you appreciate how even the smallest structures in a plant respond dynamically to their environment.